Artificial sweeteners and natural sugars may taste similarly sweet, but their impacts on the brain, particularly on glycogen dynamics, can be starkly different. While both influence energy metabolism and signaling pathways, they do so through divergent mechanisms that are only beginning to be fully understood. As more people turn to zero-calorie sweeteners in hopes of avoiding the health risks of excess sugar, the question arises, how do these substances influence the brain's energy reserves, particularly glycogen and astrocytes? This question isn't just academic. It may have real implications for cognitive performance, mood stability, and long-term neurological health. Natural sugars like glucose are the brain's primary fuel source. When consumed, glucose enters the bloodstream, triggers insulin release, and is taken up into brain tissue to be used immediately or stored as glycogen in astrocytes. During periods of heightened mental activity or low systemic glucose, such as fasting or stress, astrocytic glycogen is broken down to supply neurons with lactate, supporting neurotransmitter synthesis, membrane potentials, and synaptic plasticity. This tightly regulated cycle between glucose intake, storage, and utilization is critical to brain function, especially in regions like the prefrontal cortex and hippocampus. Artificial sweeteners such as sucralose, aspartame, saccharin, and stevia, on the other hand, are not metabolized the same way. Most do not contain usable calories, and many do not trigger insulin release, or do so inconsistently depending on the individual and the context. Some studies suggest that certain sweeteners may trick the brain into expecting glucose, initiating a partial insulin or incretin response, only to deliver no usable energy. This mismatch can lead to downstream confusion in the brain's energy regulation systems. In theory, this could impact glycogen cycling by disrupting the normal triggers for glycogen synthesis or breakdown. One possible consequence is impaired glycogen storage. If sweeteners alter signaling related to glucose sensing without delivering substrate for storage, astrocytes may fail to adequately replenish glycogen reserves after periods of high activity. This could leave the brain less prepared for energy-demanding tasks and more vulnerable to fatigue, stress, or cognitive lapses. In chronic scenarios, especially in individuals who replace nearly all dietary sugar with artificial sweeteners, the result may be subtle but persistent energy instability within the brain, an invisible fatigue that undermines memory, attention, or mood. On the flip side, certain sweeteners like aspartame or stevia may have more complex interactions with neurotransmitters and amino acid pathways. Aspartame, for instance, is broken down into phenylalanine and aspartic acid, both of which can cross the blood-brain barrier and influence neurotransmitter synthesis. These effects may alter the balance of excitatory and inhibitory signaling in ways that indirectly influence metabolic demands, including those placed on glycogen stores. Moreover, some rodent studies have shown that artificial sweeteners can alter the gut microbiome in ways that affect glucose tolerance and insulin sensitivity, even in the absence of calories. If these effects translate to humans, they could alter peripheral insulin signaling and indirectly influence how the brain senses and responds to blood sugar changes. Since insulin also plays a role in regulating brain glucose uptake and glycogen synthesis in astrocytes, any impairment here could disrupt the normal rhythms of glycogen cycling and impair the brain's ability to prepare for or recover from energy-intensive periods. This doesn't mean that artificial sweeteners are inherently harmful, but it does raise concerns about their chronic use, particularly in large quantities or in metabolically sensitive individuals. Replacing sugar with non-caloric sweeteners might reduce overall calorie intake, but if it introduces unpredictable fluctuations in brain energy sensing, it could impair the very cognitive stability and performance that people seek to protect by avoiding sugar in the first place. By contrast, small to moderate amounts of natural sugars, especially when consumed as part of a whole food diet that includes fiber, fat, and protein, can support steady glycogen replenishment and brain fuel availability. The problem with natural sugar is rarely its presence, but its excess and its isolation, consumed rapidly, repeatedly, and without the buffering components of a balanced meal. The bottom line is that both natural sugars and artificial sweeteners impact brain energy metabolism, but they do so in fundamentally different ways. Natural sugars feed into glycogen dynamics directly, supporting short-term fuel and long-term reserve replenishment. Artificial sweeteners, while calorically neutral, may disrupt the signaling systems that regulate these dynamics, potentially compromising the brain's ability to anticipate and meet its energy needs. More research is needed to explore the specific effects of each sweetener on astrocytic function, brain insulin signaling, and glycogen metabolism. But even now, it is clear that sweeteners are not metabolically invisible. They interact with the brain's energy infrastructure in complex and sometimes unpredictable ways. For individuals concerned with cognitive health, mood stability, or neurodegeneration, these metabolic subtleties should not be ignored. Choosing the right form and amount of sweetness may be as important for the brain as it is for the waistline.